Medical Geology Research Center in Nasarawa State University. And that's one of the examples that we have at the moment, right now, where we are trying to develop curriculum in these areas, as well as training or capacity building in these areas. Of course, medical geology is one of the areas that we cover. And by definition, medical geology, as you know, is a highly focused area that looks at the natural risk factors, geological materials, and geological events, natural events, and their impact on human health. But additionally, our consortium in collaboration with the International Medical Geology Association, we also foster innovative approaches to the study of environmental and occupational related diseases that the association, the International Medical Geology Association, may not have by definition in their mission. So this consortium is here to work with IMGA in looking at these areas and developing these other areas from the natural environment to occupational related diseases. So let's see, it's not moving now. Oh, okay, there we are. Yeah. So one of the areas, of course, is assessing global impacts of toxic elements, for example, in water quality and water pollution. And we do that through different types of collaborations, projects that are developed with colleagues in different countries, or again, through interaction with the International Medical Geology Association. One of the elements that we have been working with since the consortium was established is looking at the human exposure to arsenic as a global concern. And again, these scientists in the consortium looked at arsenic in different areas, from drinking water, diet related, from medical use, and even from geogenic or anthropogenic dust exposure. So those are some of the samples that the consortium addresses. One of the areas that we have developed and that you are going to be listening today is the program that brings international speakers to talk to us about their research and their programs in these areas, in these science areas that I mentioned during my first slide. So far, our international speakers seminar series have been able to host at least three presentations. One was shared with the Geo Latinas, is an organization based in Mexico, but that is international because cover South America and Central America. And with this organization, we actually were able to bring the topic of medical geology to many countries through the dissemination through Geo Latinas. But other speakers in this seminar series have been Dr. Anastasia Slovina from Bashkir State University in Russia. And Anastasia was our first invited speaker. And she talked to us about bisulfite and the experience of using the mineral in Russian medicine. Another speaker that we had in this seminar series was Patricia Ventura Garcia from the University of Azores in Portugal. And Patricia talked to us about a very exciting topic dealing with volcanoes and the health issues associated with the hydrothermal emissions from these natural events. Today, we have another exciting seminar today. We have our seminar today will be by Dr. Rufus Chaney from the USDA Agricultural Research Service. Rufus retired from the Agricultural Research Service in 2016 after an amazing career with the U.S. government and the Department of Agriculture. Rufus spent 47 years dedicated to the agricultural sciences and to the nation, to the U.S. At the Agricultural Research Service. So before going into Rufus' presentation, let me just give you a very quick overview about Rufus. Rufus and I, we have been colleagues and friends for over 30 years. It has been, for me, it's an honor to have him here today talking to us about his research, particularly on soils and cadmium, and his experiences along the years that he has been working in this particular field. Rufus is a senior research agronomist. As I mentioned, he worked for the Agricultural Research Service for 47 years. He is an alumni, a PhD from Purdue University. Rufus' research focus has been on the identification of improved genotypes and agronomic management practices to reduce phytoavailability in order to reduce cadmium and lead concentration, all bioavailability of these toxic elements in selected crops. Rufus also research focus areas has been in the assessment of potential risks from trace elements and xenobiotics in diverse soil amendments. Another area, focus areas have been on the development of metal hyperaccumulators, plant species and soil and plant management practices to phytomin elements from contaminated cadmium or mineralized soils. And he has also worked on identify the causes of iron deficiency, chlorosis in soybean and other crops, and mechanisms of iron uptake by crops other than grasses. Rufus has been elected to the Soil Science Society of America and the American Society of Agronomy, elected fellow of the American Association for the Advancement of Science, the USDA Presidential Rank Meritorious Senior Professional Award, and he has been also elected to the Agricultural Research Service Science Hall of Fame in 2013. Today, Rufus is also currently works as a consultant, risk assessment and remediation on a group that he developed or established on that particular field. So as member of this consortium and as co-founder of the International Medical Geology Association, it's really an honor for me and a privilege to bring today my good friend and colleague, Rufus Chaney, to talk to us about his research. He's going to talk to us today about human risk from soil, cadmium through rice and tobacco, sources, competitors, and bioavailability change, how bioavailability change early understandings. So Rufus, my friend, thank you for being with us today. Thank you for giving us this opportunity of having you here. And thank you for your so many contributions to this field of science. So welcome, Rufus. Let me just stop sharing.
You can hear me also or not?
Yes, I can, Rufus.
Okay,
we can.
I'll get started.
Yeah.
My focus has, for many years, has been cadmium risk from crops. Shortly after the situation was discovered in Jinsu Valley in Japan, every food scientist got a little bit worried about cadmium in food. Now, I was in agriculture, and agriculturalists grow foods. So there had to be some better understanding of the uptake, and I'll stress later bioavailability. But I'm giving you a new understanding that we have today that is not widespread enough. Yeah, I will be showing you why, in my judgment, rice and tobacco are the only crops which comprise significant cadmium risk to humans because of different bioavailability and accumulation. There have been established limits on cadmium in some crops through the Codex, the international program. Actually, the sale of some US, but also international, particularly chocolate in South America, crops are limited by either national or Codex limits. We've had, in the past, limits on sale of sunflower, flax, durum wheat, peanut, lettuce, spinach, soybean in Europe and Australia. So it can be pretty frustrating. But only subsistence rice farmers who grew their crop on contaminated zinc plus cadmium contaminated paddy soil and consumed the homegrown rice for decades have suffered the severe cadmium disease from the diet. If we don't take into account bioavailability and so on, we're going to cost ourselves a lot of wasted food and money. So let's go back and look at the fullness of adverse effects of lifetime cadmium ingestion. The first adverse effect is a renal proximal tubular dysfunction, low molecular weight proteins in the urine. If you have continued on a higher dose or for a longer time with that disease already having the dysfunction, you can have then glucosuria and amino aciduria, which is the worst kidney disease. Then if you couple the kidney disease with low calcium in diets, which rice diets provide, that can end up with the osteomalacia, which is called Itai disease. Particularly in susceptible individuals might have even lower absorption, but particularly low calcium dietaries. No milk, eat rice, you've got low calcium dietary. Repeated research has shown that high blood pressure, which is still in most of the textbooks, was never a significant effect of excessive cadmium absorption. And it's important to understand that the proximal tubular dysfunction is not kidney failure, which requires dialysis. It was never so bad as some people would like it to be. But it was horrible if you lived in Jinsu Valley, Japan, or one location in China, where the combination of low calcium diet and repeated, long life exposure to high cadmium rice, particularly the older women would have had multiple fractures. I met when I visited Dr. Higino's hospital at Jinsu Valley, Toyoma. I met a woman that had 200 fractures, if you look at her bones. She'd lost 30 centimeters of body height as bone collapse, pelvis collapse, etc. So for those who had this combination of low calcium and high cadmium diet prolonged for decades, it was a severe effect. That's also the pelvic region, fracture, and the rib cage. Every one of those white lines is a fracture. It was really sad. When they finally identified the problem and moved to help solve it, many of these women were brought into a clinic operated by Dr. Higino. Now let's deal with the issue of, so we know it can be bad. How does that happen? And how do we regulate it? Well, the first adverse effect is what we have to prevent. That's the urinary beta-2 microglobulin, which is a low molecular weight protein that goes in the urine of people with cadmium disease. On the basis of the microglobulin, you can come up with a kidney cortex cadmium limit. Now, to connect that to food gets complicated. Depends on the percent absorption of dietary cadmium, setting some dietary cadmium limit, which then we have to find a way to set crop cadmium limits. And from there, soil cadmium, either management or concentration limits. So it's complicated to go from preventing the first adverse effect of cadmium in humans and how we control cadmium in soil and farming. I need to stress that the assumptions that are usually made are just not valid. Not all crops comprise equal cadmium risk to consumers. And not all soil comprises equal cadmium risk to consumers, and that crops and cadmium risk is not linear with soil cadmium concentration because it's not linear with crop uptake. If I look at that same path from microglobulin to soil, on both sides, I've got factors that actually have to be taken into account. If you only ate high cadmium food for a year or two, it's irrelevant. Smoking, particularly usual the 20, 30, 40-year smoking, is a tremendous source of cadmium. And I'll talk about that at the end, particularly in counterfeit cigarettes. Different cadmium sources because of their cadmium zinc ratio. For instance, pea fertilizers have a high cadmium zinc ratio, so the cadmium is more available because it's taken up on a zinc transporter. Higher the ratio, the higher the uptake. Bioavailability depends particularly on iron, zinc, and calcium status, age, phytate and fiber in the diet or in the meal, oxalate from spinach and maybe a few other crops. And the phytoavailability, plant availability, is affected by many factors. The crop and the cultivar, actually we're breeding low cadmium types. Soil pH is very important, chloride in alkaline soils. Cadmium zinc ratio I'll show you about, particularly in zinc deficient crops, cadmium uptake is increased. Then the sorption ability of the soil on iron and manganese oxides. Nitrogen rate, nitrogen form, ammonia acidifies and increases uptake. Even the previous crop grown on the field affects the cadmium uptake. This is a comparison of the relative cadmium uptake in a number of crops that we sampled in a nationwide study in the United States. So I'm showing the percentile of cadmium 0 to 100. You'll see that corn and rice are down there at the bottom, and they're almost none of them go above the limit set for wheat, 0.2 ppm. But on the other hand, durum wheat is much higher than non-durum wheat, and that had to be solved, and it was solved by finding a genetic solution. Sunflower and flax both naturally accumulate much higher levels than other foods, grains, and breeding has already been done on sunflower, and it's being managed by knowing which soils produce low cadmium crops so they can make sure they market low cadmium crops for humans. So this variation of crops has to be part of your picture of what we have concern about with cadmium. I mean, how many kilograms of sunflower kernels do you eat a year? Not a lot, nor flax, but a lot of wheat, a lot of rice, particularly of rice. I want to show a picture of zinc cadmium interaction. In this, I've shown interaction with soil zinc across the top, soil cadmium on the left going down. And I showed them the cadmium zinc ratio of the metals that we added to this test soil. I show some in green. Some in green, which have an acceptable cadmium zinc ratio of 0.015 or lower. Then we have some others, the blue, where the zinc is so toxic that it actually reduces yield or kills the crop. Then we have the red ones, which denote a high cadmium zinc ratio, which might allow much higher cadmium accumulation. So if you look at the zinc and cadmium in these under these interaction studies, you see that in the top, the green all are lower than the levels of cadmium which would cause human disease. The ones which get like 25 ppm in the left bottom is killed. It's killed by zinc. So that you get to a certain cadmium zinc ratio, you have essentially total protection against excessive cadmium in your diet. So whenever the green then again denotes non-phytotoxic zinc, low cadmium zinc ratio, which gives us a safe food. This is what zinc toxicity looks like when you have an acidic high zinc soil. You have severe stunting, you have chlorosis, yellowing, and necrosis, brown and dying. And it tells you when the zinc kills a crop, you know you have a problem. But if you didn't have the zinc there to kill the crop, you'd never know you had a cadmium problem until you had dangerous levels of cadmium in the crop. This is a list of the factors which have been found to cause major variation in crop cadmium. Soil pH in particular, the more acidic, the higher the uptake. So acid rain, natural soil genesis, use of acidifying end fertilizers that make the pH lower and cause more uptake. Crop species different, I showed you, and cultivars different. In alkaline soils, but not acidic, high chloride matters. So potatoes in Australia grown with chloride-rich irrigation water had much higher cadmium than potatoes grown without that high chloride. Soil zinc, low zinc increases, high zinc reduces cadmium. I showed you that. Sometimes biodegradable organic matter, too much organic matter can increase the uptake. Previous crop, legumes cause higher uptake than previous crop of grasses. The higher the rate of end fertilizer, particularly ammonia type fertilizers, the higher the uptake. In alkaline soils, even the rate of potassium chloride fertilizer can affect the uptake. There are some situations where there's aerosol cadmium emissions near the field, and that can cause high leaf, but not necessarily high grain. Then we have the geogenic types. There are a number of different geogenic sources. Some call them Allen shales, some marine shales. There's a number of different unusual kinds. Some of the worst are phosphorites that came from bird dung from millions of years ago. I'll show you a little bit more about that in a minute. And then, of course, there's the geogenic mine wastes from zinc, lead, copper, and silver ores. When those are dispersed, but at least the cadmium zinc ratio is not high on those. Here's a picture of natural mineralization with cadmium relative to zinc. We have in Central California an area that has soils derived from marine shale. And instead of having less than a half ppm in the soil, some of those go up to 2 and 3 in farmers' fields. But I've done research on soils, farmers' fields with 9 ppm that cause very high cadmium in the crop. Now let's look at Jamaica. Here's our 4 elements, aluminum, zinc, cadmium, and across the whole of Jamaica. And then in that central province, Elizabeth and Manchester provinces, it shows extreme. There you go above 95th percentile of cadmium concentration in soils occur in those red dots on the right bottom. Those are totally natural from soil formation processes that happened millions of years ago. But now are occasional sources where the cadmium is higher than the zinc, which is nobody else has found anything quite that bad in the rest of the world. But you know, that's natural processes. In Europe, they've had a EU-wide soil survey program that was done with exceptionally good techniques. And so this slide shows their slide of the distribution of cadmium in European soil. You see there are some that are up to 5 ppm, natural rather than contaminated. Then they also made a map of cadmium zinc ratio. You'll see that they have some of these marine shale, alum shale, whatever you want to call them, they exist, and they provide the same kind of potentially cadmium risk if they're acidic in particular. If they're alkaline, there's some of those in the bottom might be alkaline enough that there's not a lot of cadmium uptake. But those that are acidic and high cadmium zinc ratio, they can't avoid it. And here's the one that I've been working on for the last 3 years, is the cadmium in chocolate. In South America, there are a number of, let's say that nearly all the soils are too acidic. pH is as low as 4.5. But in locations in at least 6 countries that have been evaluated, there are these high cadmium zinc ratio soils, which cause potentially then much easier transfer of soil cadmium to cocoa beans. In Colombia, this one Santander Valley has many of the red dots, which represent greater than 10 ppm, and the highest was 27 ppm cadmium in a natural, uncontaminated soil. That's it. We got to keep that in mind. These are not contaminated, they're mineralized. Now I want to change to stressing about rice. Actually, rice is the only dietary source which has actually caused human disease. If you think about it, there have been lots of other worries and claims. For instance, they thought in Sri Lanka they had a cadmium problem, but it was shown that it was bad analysis and interpretation. As it happens, rice transports very little iron or zinc to the grain, and milling the brown rice removes most of the zinc and iron. And even on highly zinc cadmium contaminated soils, the grain zinc remains very low. Rice, different from every other studied crop, handles zinc and cadmium differently, and I'll show you more about that. Now try to imagine in Jinsu Valley, Japan in 1970, the fact that the subsistence rice diets where they ate homegrown rice would then have been deficient in iron and zinc, and actually also in calcium because they didn't have major sources of calcium in their diets. And that would promote the cadmium absorption, and that did become a major public health problem. So the rice case is special. Other diets with high cadmium have not caused human disease. Garden crops, wheat, shellfish, sunflower kernels, I'll show you the low bioavailability limits absorption. But those subsistence rice diets have high bioavailability. And I'll show you more about that because we should be regulating bioavailable cadmium, not total cadmium. The bottom point is that livestock exclude nearly all feed cadmium, only accumulating cadmium in liver and kidney. If you don't eat the liver and kidney, you don't get cadmium exposure. Here's one example of that from oyster eaters, oyster fisher persons in New Zealand. They happen to have an oyster that had high levels of cadmium, of 5 ppm or more in the oysters. And if you can imagine, if you're fishing them, you might eat a lot of these delicious oysters. And so they were eating from none to 10 oysters per day on average. And you'll see that there's a low slope of blood increase in cadmium of non-smokers and a jump to smokers. The fecal cadmium line shows the people were eating the cadmium that they were claiming they were eating. So we have here an example that there's very low bioavailability of cadmium in oysters eaten by people that eat a lot of oysters. Another study was done of young women in Sweden who either had normal diets or ate shellfish as part of their diets. The cadmium in the crop in their diets was about 2.5 times higher for the shellfish consumers, but the blood and urine cadmiums were not increased. So the shellfish cadmium was not bioavailable, caused no increase in risk. Same kind of study we did in North Dakota with sunflower kernels. Fed them as once a day a little package of snack peanuts or sunflower kernels with very low cadmium peanuts and moderately high cadmium kernels. You see that the crop was higher. The diet was about twice as high in cadmium. It showed up in the feces. The blood was very slightly, but not significantly higher. And the urinary cadmium and the indicator protein NAG were not significantly higher. Again, saying they ate all the increase in sunflower kernels, a heavy snack dose of sunflower kernels, and yet it caused no problems. I'll show you this from another point of view, and that is I did a lot of work on biosolids trying to set regulations in the United States to protect the safety of food. In one of my studies, I was able to grow Swiss chard, a spinach-like vegetable, and that was fed to guinea pigs for 80 days. So it's a good chronic feeding study. You see, I had 3 different biosolids there, 1, 2, and 3. And the pHs differed, and the cadmium and the biosolids differed. But in the chart, you'll see that only one of them, biosolid 2, caused a significant increase in cadmium in the crop. But look at the zinc. The zinc was highly increased in those 2 with the higher cadmium. And if you look at the kidney and liver, again, 80 days feeding at 28% of diet, and the kidney is not changed and the liver is not changed. We did the same kind of thing with lettuce, but I'm not going to show that for time. I then had a student who grew radioactive intrinsically labeled cadmium in lettuce and spinach and fed those to Japanese quail with the Food and Drug Administration researchers. And you'll see that when we grew zinc into the crop along with the cadmium, the absorption was reduced. And so that another picture that the zinc in the crop can either serve as a kill the crop before it has too much cadmium, or can interfere with the absorption of cadmium. Now I'm going to switch back to rice. And this is Jinsu Valley, some of the earliest data from the Toyoma area there. On the left is the cadmium in the unpolished brown rice versus the concentration in soil. You'll see that it's all over the rice. It goes, from up to 4 ppm. But if you look at the zinc on the right-hand side, you see that it reaches a plateau. It doesn't go higher than about 30 ppm. So that there's a disconnect between cadmium and zinc in rice. Now I want to show you a site in Thailand that I helped discover as being high in cadmium. This is the top of the mountain where the irrigation water enters the top of a levee series of rice paddies. And so the rice paddy at the top is contaminated by fine particles suspended in the river water from a mining area upstream. And as you go down this hillside, the concentration of cadmium gets lower and lower. When it gets down to the valley floor, it's much lower than on the hillside. But they grow a lot of rice. The rice is delicious. But when you look at the cadmium concentration, look at this. We had one soil was up to 225 ppm cadmium at the top of the mountain. But down here at the bottom, where there's not as much cadmium, where it's more acidic, you can get up to 3.5 ppm cadmium. So that the drainage and pH and so on are all involved. But when you look at the zinc, again, no change with increasing zinc up to over 6,000 ppm in the soil. It has to do with flood and pH and chemical forms. On the other hand, if you grew Chinese cabbage on that high zinc cadmium soil, you ended up with significant cadmium and zinc, but you got both. Now I want to move on to the absorption of cadmium in animals, because bioavailability should be part of our consideration whenever we're talking about regulating or protecting against cadmium, geology, medical geology. These studies have found that for some people, there's unusually long retention of cadmium in the duodenum, long after the other meal constituents were excreted. They're absorbed into the cells, and then slowly, as those cells are exfoliated, they enter the feces again. So Fox had shown that, and then Shaken-Smith showed. I'll show you a slide on that in a minute, and then I'll show you work we did with rats. So here's the Shaken-Smith data. Human volunteers ate 109 cadmium labeled meal, and you'll see that it took days before the body total cadmium equilibrium was reached. In our feeding studies, my colleague made polished rice. He bought polished rice, actually from Thailand, and it was cooked, freeze-dried, and mixed with the complete diets of 40% dry weight percentage of the diet. The background diet was less than 0.01 ppm cadmium. So when he added the cadmium spike to it, isotope, it was 0.24 ppm. He was a world rank nutritionist, and he knew how to grow rats with a marginal supply of iron, zinc, and calcium that did not reduce their growth rates. Mild deficiencies, but no reduction in growth or health. The females then were fed marginal or adequate level diets, 35 days, fasted, fed 1 gram of the labeled diets, and then returned to varied diets up to 64 days. And I'll show you the kinetics of that. We are particularly looking at turnover in the intestine of cadmium from that meal, and then of course, liver and kidney. Our first studies were with sunflower kernels and rice, and then these diets with low zinc, low iron, low calcium. You look at those, you'll see that if you combine multiple marginal supplies, you have much more absorption of cadmium from the sunflower kernels, and particularly from the rice, up to 10 times higher absorption from what I would call a normal low zinc, low iron, low calcium rice diet compared to a normal diet with sunflower kernels added. So when we did the 64-day study, here's the way my colleagues like to picture it so you can see the kinetics. Nice, smooth kinetics with a slower loss from the marginal animals. I like to look at it this way. This is the difference in the area under the curve is the extra absorbed cadmium from the marginal diets, remarkably increased. Over time, it goes into the liver and then goes out of the liver and ends up in the kidney. So that you see absorption, a nice logarithmic pattern that we see, but now marginal intestine causes much higher uptake. If you thought it was metallothionein, we have another paper about that, which I'm not going to discuss, which showed that under the conditions of these animals, metallothionein binds no cadmium whatsoever in the intestine. Now let's look at the Japanese case again in that Jinsu Valley where the Itai disease was discovered. I'm showing here the elderly patients who were sampled in some of the original work there. Look at the urine cadmium, where 10 is a cutoff for maximum that could be possibly safe. You see that they were well above. And then look at this beta-2 microglobulin in urine. Average 91,000 to 91,000, 102,000. Even for the people that didn't have severe disease, that was above 5,000, up to, again, 16,000. Whereas controls not in the valley had the usual less than 300 and urinary cadmium less than 5.7 in Japan. So that the effect of eating that marginal diet with high cadmium caused disease indicated by urinary proteins. Other work in Japan supports the idea of a 10 micrograms cadmium per gram creatinine threshold before you start to have the extreme cadmium, beta-2 microglobulin urine, both men and women. Others in Europe want you to think that disease starts at 3 micrograms per gram creatinine. But here we have a naturally exposed population that clearly do not support that point of view. Now let's come back to how do we deal with rice and cadmium. Sophisticated epidemiologic studies, particularly by Nogawa and Nokeda, indicates that the 0.4 ppm limit for homegrown rice in Japan may be a little bit high for homegrown, 100% homegrown rice. Codex has a 0.2 ppm. No, Codex has 0.4, but EU has 0.2. China has 0.2. But I think we should keep in mind that these people that suffered disease had poorer nutrition than farm families in Japan and China today. Today they have meat and they have more balanced diets. Ikeda did some studies examining cadmium in diet, blood and urine, indicator proteins of cadmium disease, studied over 10,000 Japanese non-smoking urban women, middle-aged, 30 years adult exposures. They found no person with any evidence of renal disease from cadmium. Further, all were less than 3 micrograms per liter in urine. So the claims about high risk from to a Swedish and Belgian citizens from lower amounts of cadmium have to be taken into consideration, all of this Japanese and Chinese data. Yes, some horrible things happened, but think of World War II and the poor nutrition that these people were eating high cadmium rice. We can understand better when we think about the history of their exposure, why they had more severe disease. So let's look at populations exposed to high dietary cadmium. I showed you the villages in Japan and China, rice grain, subsistence rice, lots of people with disease. In Bluff, New Zealand, oysters ate hundreds of micrograms a day, no disease caused. In Shipham in the United Kingdom, gardens were grown on mine waste, gardens up to 300 parts per million cadmium, but they were alkaline. And there was no disease in any person. In Stolberg, Germany, another area with mine waste and smelter contamination, 100 cadmium, 10,000 zinc. Garden foods were grown there. No adverse cadmium health effects in long-term residents. In Palmerton, Pennsylvania, in the US, there was a smelter that contaminated gardens up to 120 ppm cadmium, 10,000 ppm zinc. But because the emissions were alkaline, the soils were usually alkaline and there was no zinc toxicity. Garden were higher than background crops, but their EPA did a full human survey and found no individual with high cadmium due to the gardens. Whereas smokers had high cadmium, not disease high, but high. And then this is the picture of cadmium absorption from feed and livestock fed a relatively high cadmium crop, in this case, corn maize silage, grown on biosolids amended soils where the biosolids had more cadmium than are legal in the United States. This was in the early research days. So if you look at that, you'll see low, medium, and high. Come over to feed cadmium. In 3 years, these goats were fed 3,900 milligrams of cadmium versus less than 40 in the controls. You see the livers are very slightly increased. Kidney was increased somewhat. Muscle not affected at all. And the liver, the total amount in milligrams of cadmium in liver plus kidney was only 1.4. So it's 1.4 out of 3,900 milligrams of cadmium ingested by the goats ended up in edible tissues. The muscles were not detectably increased in cadmium. So keep in mind how protective the normal cadmium zinc ratio can be of any accumulation, particularly in livestock, but also in crops. Oh, sorry, wrong one. So again, how did we solve all the questions about biosolids use in the United States? EPA imposed retreatment standards, and the biosolids have to have extremely low concentrations, much like EU. Retreatment solved our problems. So it took enforcement of regulations in order to have biosolids that could be used. And then the biosolids are regulated as well. Nowadays, the annual rate of biosolids is limited by the phosphorus fertilizer, which couples it to a very low cadmium addition. The inorganic fraction of biosolids, not just the organic, ends up binding cadmium strongly. And because now we've limited cadmium loss and zinc is used by people, the cadmium zinc ratio in biosolids is much lower. So the cadmium availability to plants is much lower. Further, nearly all those are used for grains, which exclude most of the cadmium in the first place. So we think we have a quite safe program for biosolids use, recycling nitrogen, phosphorus, etc. But it's done based on a great deal of research. I did a number of studies on long-term biosolid soils. Some of them have been amended for 30 or 40 years. And we then did an experiment using stable isotope cadmium added at 0, 2.5, 5, 7.5, and 10 milligrams cadmium per kilogram of the control or amended soil. They were all carefully adjusted to pH 6.5 and 0.1 molar calcium nitrate. Proper fertilizers grew romaine lettuce to maturity. Here you can see what it looks like. On the top, our T28 was 700 and some ton per hectare of that particular composted biosolid. You'll see that in the bottom, the control, there's some chlorotic leaves with a high cadmium. But otherwise, you don't see the injury from the metals. Now look at this. This is the beauty. The red line is the cadmium salt added for the controls, no biosolids. Nice high slope up to higher cadmium than we'd ever want to see. But if you look at the light green line for the heat-treated biosolid, applied at 224 ton per hectare, it's linear, but its slope is reduced. And then with the one with 672 ton per hectare of the compost that only had 7.2 ppm cadmium, even lower slope. And now most of our biosolids are under 2. So we're down there where even when you do it in the worst possible way, you're either going to have a dead crop or low cadmium. You can't have high cadmium healthy crop. Now I'm going to introduce a whole nother line of understanding, and that is rice versus spinach. In the last decade, we've seen discovery of uptake of cadmium by different crop plants. rice was discovered to use an unusual transporter, a manganese transporter called NRAMP5, whereas spinach uses a zip transporter in the roots to take up from the solution. So what we did was I developed a nutrient solution system with controlled chemical activity of zinc and cadmium, so that when I buried the zinc, I had no change in the activity of cadmium. You see that both of them went up to above log 3, so that's above 1,000 ppm of zinc at the highest, and they had reduced yields. Then shoots and roots, both spinach and rice. But here's the beauty. On the left, you'll see that with rice, which uses a manganese transporter to take up cadmium, the green, the shoots, the cadmium in the shoots was not reduced even at the point of zinc phytotoxicity. On the right, with spinach, you'll see that with a little bit of added zinc, the cadmium in the shoots of spinach goes down, and it goes down and down and down with further, and the roots go down after 10⁻⁷ molar activity. So again, it shows you that there's fundamental difference. And we don't know other crops that use the NRAMP. We know that everything else has been studied uses a zinc, zip transporter for zinc. So this helps you see that you can exclude zinc from rice, but not spinach. My colleague Aro in Japan did a study of, again, the flooding versus aerobic periods of growth of rice. In continuously flooded, the soil has a high pH. The dissolved manganese is very high. The dissolved zinc and cadmium is low because of the high pH. Well, people are trying to drain the fields more to reduce the arsenic uptake by rice, which is a natural phenomenon. So then if you drain it a little bit earlier than continuously flooded, you get a little bit. But if you drain it at the beginning of heading or at the whole growth period, you can have much higher cadmium just because you drained the field. We did a test, again, because they call it alternate wetting and drying, flood management, is important to lower the arsenic in rice grain. So you'll see on the left, the arsenic, as we go from full flood to dry, arsenic gets lower and lower. But as you go on the cadmium on the right, you go from flood to fully aerobic, the cadmium increases even faster than the arsenic decreases. And so you can't just use flood management to reduce arsenic without thinking about cadmium. I want to show you why we know it's the NRAMP transporter in rice. This is data from Ishikawa. They did, you can imagine if you're Japan, you put a lot of money into understanding cadmium in rice. After doing all kinds of other studies, they decided they would do mutation breeding. They used ion bombardment to make mutants of one of the main favorite rices of China, and then grew them out in the field and measured the cadmium in the grain of every plant. And they found 3 different genotypes that had remarkably low cadmium. And then in this slide, they grew one of those low cadmium genotypes in 3 different fields with high cadmium in the soil. You'll see that where they got up to 1.8 ppm in that middle one, the control, that is the mutant rice, had 0.02. So they then developed, because it's legal, not a GMO, they developed a cultivar that is a low cadmium of this favorite food type of rice in Japan. But I think it's pretty clear that it's NRAM5 and not something else. They also discovered a root protein that pumps cadmium into the vacuoles, which could stop cadmium going up the stem toward the leaves and grain. And if they overexpress that protein, they could stop cadmium translocation. But that, so far, they haven't worked out a non-GMO approach, but they're looking to do that. And in China, where they really need this kind of genetic quality rice, they're investing a lot of research on that. I want to show you again that manganese cadmium. This is an experiment where manganese was supplied at different levels. And you'll see that when they supplied it to the wild type, the uptake was reduced. Whereas on the mutant, the added manganese had almost no effect. And that's again because the transporter was knocked out and it didn't take up manganese, and therefore the manganese couldn't inhibit the uptake of the cadmium. So we have a lot, you know, when there's a real problem that causes human disease, study it and you'll learn how to fix it. Unfortunately, one of the things that's not being fixed and is international as opposed to only on these rice countries is cadmium and tobacco. I show you here tobacco grown on 3 biosolids amended soils back in 1970 when the biosolids were quite contaminated with cadmium. So you see that the cadmium in the bottom one got up to over 3 ppm in the soil, DTPA cadmium. And this cultivar, they prefer it to be grown in strongly acidic to protect it from root diseases. You see that the cadmium in the tobacco is much higher than is preferred or even tolerated. But we don't regulate cadmium in tobacco. Don't you have to wonder why we don't regulate cadmium in tobacco? Unfortunately, besides the ethical manufacturers, if you can call them ethical, of cadmium, they keep the cadmium in cigarettes about 1 ppm dry weight. But when we look at counterfeit cigarettes that are being sold a number of locations, we did a survey with a student group from New Jersey or New York, where the average is 4.8 ppm cadmium, not 1, and they went to 6.5. I know of one location where unregulated biosolids, 70 ppm, another unregulated near a lead smelter, 2.6 ppm cadmium, and one grown near a zinc smelter in another city with 25 ppm. They all want to sell their tobacco. So they sell their tobacco to these people that produce counterfeit cigarettes where nobody can regulate and it's hard to find them and put them in jail. So what have I told you? What have I helped you understand, I hope? The most important take home is that rice is different than other staple foods. It excludes zinc. Cadmium is highly bioavailable. Further, that the rice diets cause iron and zinc and can contribute to calcium deficiency and subsistence, homegrown rice consumers. And it comprises a major health problem, and that China has tens of cities with that kind of a problem. The marginal supplies of iron, zinc, and calcium promote cadmium absorption into the intestine and retention into the kidney and liver. With adequate supply of zinc, iron, and calcium, the cadmium goes into the mucosal cells, but it's excreted, and much less goes into the storage in the kidney. There is a high absorption cadmium and long turnover in the duodenal cells, which allows a higher net absorption into the kidney in the end. Because those rice subsistence diets made them susceptible, rice cadmium is absorbed more effectively than other foods. If other foods contain the same increased cadmium with 100-fold zinc, they would have had much lower absorption. And we showed that with animal feeding studies. I argue that all food standards, and particularly Codex standards, should be based on the bioavailable cadmium rather than total. Yes, it's easy to measure total, but research has helped us show what makes it more bioavailable, and we can make better science-based rules than we have today. But certainly any limit should reflect the presence of increased zinc along with any increase in cadmium, any iron, and so on that affect the potential absorption and risk. We have to remember that zinc and cadmium are a pair, except in rice and flooded soils. These other foods don't have low zinc and iron, and they've never been shown, except rice, to cause this high absorption. We have a number of what we would call minor foods: sunflower kernels, flax, poppy seed, and chocolate, which can have higher than background natural cadmium levels, but have zinc, have iron, and they're only eaten in small amounts. So it says that, to me, that sources of cadmium which lack 100-fold zinc may comprise higher risk, and we should focus our regulations on that situation. If you want to have zero increase in soil cadmium, as some seem to think is necessary to protect humans, we couldn't have any phosphorus fertilizer except electric furnace product, much more expensive. Livestock feedstock stuffs should be grown only on the farm where the livestock were raised. And then you're recycling the cadmium from your soil back to your soil. If you import grain from, say, the US or Brazil, you're adding cadmium to your soils. If you want zero increase in cadmium, you can't use any biosolids or MSW compost. And those are generally seen as proper environmental management today. And you would avoid high chloride land for any human food crop. You'd have to prohibit and actually stop all tobacco. You'd believe that shellfish and liver and kidney consumption would be dangerous to you when it's not, by any data anywhere in the literature. And the consumption of flax, sunflower kernels, durum wheat, and chocolate, again, there's no evidence. We need more bioavailability research, yes, but we don't have data that says that we have a risk from those only through rice or crops grown on high cadmium zinc ratio soils. Where has improved science affected needed regulations? One is the diagnosis of induced renal tubular disease. There were lots of, let's say, concerns about beta-2 microglobulin, more recently shown to be within the normal range. The lack of appreciating the role of the subsistence rice diets in causing higher cadmium risk. And again, the other feeding studies certainly contrast with the rice feeding. And it's just the natural exclusion of zinc and iron and calcium from rice grain, natural. No other crop is as high a risk in my experience. So in rice, no co-contamination with zinc because the transporter is the manganese transporter, not the zinc transporter. Many studies of cadmium uptake use maximum tolerated dose, 50 ppm in the diet, not 0.2 or 0.4 that are like the human diets, or 1 ppm. When you feed 50 ppm, you screw up everything in the intestine that affects absorption of cadmium. So it's just artificial studies and artificial conclusions. When we feed crops, we get the relevant bioavailability. Okay, I've told you my story, and I'm ready to work with Jose to deal with any questions that people have.
Thank you, Rufus. This is fascinating research and highly relevant to many areas that we actually cover and that we need to work with. I am really interested in a couple of issues that you mentioned, particularly that disconnect that you described between cadmium and elements like zinc and iron, because this is something that could actually be fixed in terms of supplementing soils, I would say, with these elements. But that's a very, needed area of work as well. I do have a couple of questions that I would like to ask you before we open the presentation to other questions. Oh, by the way, I would like to let people know that please, if you stay for the next section, which is questions and answers, we are going to have kind of a panel, and then we are going to open this to everyone that would like to ask Rufus questions about the presentation and the material. Rufus, I would like to take the first turn on this, and I would like to ask you something about your what you discussed about the regulation. I worked for the FDA for quite some time, looking at regulatory science, but applying mostly to medical devices. But as you know, the FDA has this center, the Center for Food Safety and Nutrition, and I'm not quite sure if that center actually has any regulation published dealing with cadmium in rice. Do you have any do you know of anything that has been addressed by the FDA in that particular area?
I know the people at FDA that work on that issue, and so far they haven't discovered in the U.S. market high cadmium rice. I'm surprised they haven't found any, considering that Thailand and China ship rice to the United States to sell. Yeah. But they did discover that in those California soils I showed that were mineralized with cadmium, the marine shale. In 2004, a batch of frozen spinach had 40 ppm cadmium dry weight, whereas the EU limit would be 2. They forced that removal from because it's frozen, they can make it be thrown away. And so they did regulate that one, and that forced the growers to stop using those high cadmium soils. The Food and Drug attempts to prevent the problem more than develop a numerical regulation.
Well, I know that they have been recently, in the last few years, they have been working on looking, for example, at arsenic in rice, and they have developed a few regulatory aspects dealing with arsenic in rice, looking at mostly at inorganic arsenic and,
inorganic,
and this kind of thing. But it will be, I think, based on all this fantastic data that you have presented, it will be, I would say, a good approach for the agency to look also at the cadmium issue. Not only, I would say, not only at from the food perspective, but also, there is another center at the agency, that is Center for Tobacco Products. And based on what you described today, that has to be one of the concerns and issues that other centers should be looking at, particularly for cadmium in tobacco, which I don't know if it has been addressed by the agency.
Not tobacco, as far as I know.
Yep.
Other than helping scientists show that these counterfeit products are contaminated. Let me explain a story about arsenic in rice in the U.S. and industry response. Everybody agrees that baby cereal is the most sensitive potential arsenic transfer from rice. And so the manufacturers said to the rice industry, we will only buy your rice if it has lower than this much arsenic. The only way to grow it with that much low arsenic reliably was to use more aerobic soil conditions. And so they made contracts to sell low arsenic rice. And so all of the baby cereals made in the United States are low in arsenic, inorganic arsenic. We were very lucky in the US that the soils where rice is popular now, good yielding soils, were not ever cadmium enriched. so that we don't have a cadmium problem in our rice from California or the South Central US.
Mhm.
We were lucky. So industry has a way. You tell me you make it on television and in newspaper.
Mhm.
They're going to find a way to not have that show up again.
Mhm.
And I helped work with the industry, developing a very inexpensive method of analyzing inorganic arsenic, so they could do it at a reasonable cost. The full arsenic speciation of one sample of rice was $150.
Mm.
Mine could be done for less than 20. It
Uh-huh.
uses standard ICP AES techniques for arsenic.
Uh-huh. Mhm. Mhm. Actually, Rufus, that is a very interesting aspect. Also, what you just mentioned about arsenic in rice, because there was a point in your presentation that also brought a lot of concern to me about the understanding of this issue, of this area. And what I'm referring to is when you mentioned about the flood management
Yep.
and this technique that basically what you mentioned there is that it tends to obviously the purpose is to reduce arsenic in rice,
Yep.
but that approach from your data shows that has the resulting issue that increases cadmium. If I
But
s- if
it's a marginal increase, Jose, so
uh-huh.
that it's still within the safe concentration for human consumption for a lifetime.
Oh, I see. Okay. So
Because they by luck, they were not contaminated or mineralized in the history of Earth.
uh-huh. Okay, I see. So it brought me. I saw kind of a linear increase in cadmium.
Yeah.
So it brought me a little bit of a concern into that
Yes. Yeah.
because it's not monitored. It's not actually looked into in the U.S. So that's something that that needs to. I thought that it would need to be addressed as well. My final question. My final question.
When you think of Bangladesh and India, where they
Uh-huh,
grow a lot of rice and huge rice diets, the soils are alkaline. So if they use aerobic to reduce arsenic because they're polluted with arsenic,
uh-huh.
they don't have an increase in cadmium because it's controlled by the pH.
I see. Yeah. My final question to you, Rufus, and then I would like to open the floor to everyone that would like to ask questions. Your data on cadmium and tobacco is fascinating. I mean, I a long time ago I did some work when I was working with the Armed Forces Institute of Pathology. I looked into cadmium as a element that with time, this cadmium accumulation in the prostate is an issue. It's related to prostate dysfunction. And at one point, cadmium was in prostate tissue, was used as a marker, as a clinical marker for prostate to look at a prostate issues clinically. So in fact, I published a paper where we have archival material from smokers versus non-smokers,
Oh boy.
and the cadmium levels in smokers was incredibly high, and selenium levels were very, very low. So there was kind of an antagonistic effect there because apparently the cadmium replaces the selenium. Selenium is a micronutrient for the prostate. So for healthy prostate, but for smokers, selenium levels were basically deficient, very deficient in the prostate versus cadmium level were incredibly high. So the concern here is, I know that there is a clinical issue here, but the concern here is that, cadmium, like you mentioned, is not regulated in tobacco. And now with this cigarettes, what do you call not the these cigarettes that are designed apparently to reduce the habit of smoking, but actually people smoke more. What they call it? This it comes with the battery, all that. So there is no regulation at all in those products. So your observation here that cadmium increases or is high in counterfeit tobacco that is used in counterfeit cigarettes, it should be addressed by the FDA. So
let me describe another recent study. A young man worked for a company where he sprayed cadmium paints. So he had to have, OSHA requires cadmium analysis of blood every month or something like that. And all of a sudden his went from like 3 to 6 or 7 and violated the 5. And so he had to be taken off work with cadmium. And they kept monitoring him monthly and it didn't go down. It didn't go down. So after a couple of didn't go down days, they said, well, they interviewed him. And they finally found that he had started smoking cheap counterfeit cigarettes. As soon as he stopped smoking the cheap counterfeit cigarettes, his blood cadmium came down and he was able to go back to his regular work. And the work didn't cause higher exposure because OSHA has lots of protections built in against exposure at work. So that just smoking those, and they were not the worst ones. They were only about 2 ppm, not the 5 and 10 ppm. So I think that not dealing with cadmium and tobacco is a serious loss in our countries.
Right, right. So Rufus, I would like to thank you for answering my questions, and I would like to open the talk to the rest of the participants here. We have questions from Munir Seal. His question is, Munir is from Pakistan, and he's a highly interesting talk, as always. Any comments about the EU report 2019 that mentions there is a risk from cadmium in phosphorus fertilizers, I suppose, if the analysis value is beyond 20 ppm. Munir, if you want, you can also establish your question as well, or mention, ask Rufus as well.
Hi, Rufus.
Hi.
This is Zia. How are you?
Yeah, I saw your name there, yeah.
Since I work for a fertilizer company, so I am more interested towards cadmium limits in fertilizers.
How much do you have in your products?
We all import from Morocco.
You're going to have to eventually, except you have the advantage of alkaline soils, so that it's arguable whether the EU limits are so necessary. Let me say something about the EU limit.
Rufus, can I ask you something very quick? Could you stop sharing the screen so we can see the rest of the audience?
No come.
Are you there, Rufus?
I'm here, yes.
You might want to stop sharing the screen so we have the we can see the rest of the audience.
Okay, let me answer the question you asked, which was about limits in fertilizers. In Europe, they've done what I think is appropriate. They've made a regulation based on milligrams cadmium per kilogram P2O5, because fertilizers can vary from almost no phosphorus to 40% phosphorus, 36% phosphorus. And so the regulation should be based on the ratio of cadmium to phosphorus, because that's the source is only the phosphorus part of the fertilizer. There's still argument in Europe whether it should be 20 or 40 or 30 or 25. But certainly it's lower than the very highest. I'll point out that the reason that Australia and New Zealand have problems with cadmium because they used some of the Christmas Island phosphate, which was quite high in cadmium. Used it for decades and decades before anybody knew about cadmium. Go ahead.
Any other questions for Rufus? Mm-hmm. Okay.
I couldn't understand if there was a question asked there.
I thought that Teresa had a question, but I could not, yeah, because I'm not seeing the screen right now, Rufus. So. Okay. Jesus, do you see if anyone has questions?
Not yet.
Okay. Teresa, do you have a question for Rufus?
No, I have no question. I was for a moment on my PC and my cell phone because with a handy, but
Okay.
I find it was really very interesting, and it's a very rich presentation because all the problem of the interactions of the different minerals and metals is something that we study in some of our courses with our students. The Dietary is a case which we always treat with our graduate or some postgraduate students. It's it was a very important experience. And Uruguay, there are other problems because each country has a different soil, and here are other interactions with copper and selenium, and this is always an important topic for us for the toxicological effects or the problem of scarcity. When something is absent, it's also that is a typically, there we always explain to the students the difference between a toxicology effect and another health problem. Because in toxicology, it's because there is too much, but very often if there is too little, well, it's really a very rich study. Thank you very much. We will go through it later.
Yeah, let me comment about that. We know that there was soil cadmium into rice, into people. In Japan, there were 52 villages with the disease. In China, I've never seen a number, but it's probably hundreds of villages because they had extensive zinc lead mining and lack of regulation in communist countries. And so many rice paddies downstream were polluted by smelters and mines upstream. Now they're working very hard to solve that problem. And we think the mutant rices with little cadmium uptake may protect the people. But I've seen tobacco grown in China with 40 ppm cadmium, and really anomalous and unacceptable. But it's again, it's part of that counterfeit tobacco business. I think if everybody takes home that understanding of the competition starts in the soil and moves finally to the small intestine. And how do you deal with that complexity? Well, you have to deal with the soil contamination very strongly. Then you have to find a way to deal with marketable crops. And these rice problems, certainly we understand now that we have a very different level of risk from wheat and soybeans and other foods. Interactions are everything. Bioavailability is everything.
Rufus, one other very interesting area that you mentioned was cadmium in chocolate. There is a lot in that we don't really know in that area, and that's a very significant industry, you know, or in South America, for example.
Even in Puerto Rico.
Yes, here as well. Yes. So the that, you know, if is that regulated in a sense, or is that actually, if there is anything that is being applied to look at the cadmium levels?
Yes. The EU set in action in 2019 an absolute limitation on cadmium and cocoa beans entering Europe at I think it's 0.8 ppm, and in the chocolate produced. So it gets complicated because the shell of the cocoa bean is higher in cadmium than the nibs, which make the chocolate. But in the process of working with these, I lost you.
No, I'm
Okay.
Yeah.
Okay. The cadmium level in cocoa beans and soils has been measured in a number of countries, both in Asia and in South America and the Caribbean. And unfortunately, the ones in the Caribbean and South America are usually very strongly acidic, which promotes cadmium uptake. And then in several of the countries, I helped them find a subset that have high cadmium zinc ratio. These cadmium mineralized marine shale, alum shale kind of natural high cadmium soils. Some of them have a little bit of mine waste contamination. You know, the Spaniards came over a long time ago and spread mine waste all over. But that's a very small part of the cocoa production area.
Okay.
I've served on a committee sorting these things out for a California court. I know a lot of things I can't talk about, but I can talk about the published data. And the published data show us that, like that valley in Colombia that I showed, had 27 ppm cadmium.
Wow.
And that's right up there pushing Jamaica. And it's just a natural process, a natural phenomenon, but it means that you can't grow cocoa there. You can't grow spinach there. Maybe you shouldn't even grow corn there. They haven't looked at other crops because this is a tree, you know, a forested area with slopes that you don't really want to grow other crops on. So they're happy to have the coca rather than cocoa plants.
Yeah. Another thing, Rufus, is that you know, unfortunately, many of the regulatory aspects that we know about toxic metals or elements in food has been established based on the levels, the concentrations of many of these elements, many of these toxic elements. There is the only one that I know of that has been established, the regulatory aspect that have been established based on the form of the element or based on the speciation of the element is for arsenic.
Right.
But we are more concerned about the inorganic. But your point about the form of the element or based, yeah, your point about the issue that it's very important to consider the bioavailability. That has not been in any way considered in the regulatory side of this issue.
Yep.
So
I agree.
How do you suggest that should be established? Because that will require the industry to do a lot of different type of assessment and measurements and experiments in order to comply with that.
I tried to explain my view about that in the summary part of my talk. I think that we should rely on the available data. Then if a lettuce or spinach cadmium limit was to be developed, it should consider the presence of zinc in the crop and iron availability in the crop, the things
Right.
that nutritionists know about pretty well.
Right.
In the case of rice, we know that separately, there's been an effort around the world to develop rice and corn and other staple foods with higher levels of bioavailable iron and zinc because of deficiencies in subsistence populations around the world. So they might be able to double 50% or double the zinc level in rice, polished rice. That's still not going to be enough to inhibit cadmium uptake. But the same soil would grow soybeans and wheat and corn and spinach that would be perfectly safe for a lifetime of consumption. The food regulators need to take into account bioavailability.
Yeah.
They don't like them.
Yeah. For sure. That's for sure. Yeah. I think, Teresa, you have a question concerning what? If there are any studies of cadmium in coffee or tea? Is that your question, Teresa? You're on mute.
Yes, that is my question. Yes, because when you were speaking about the chocolate and the meaning it has, I mean,
we're lucky in that chocolate is like spinach, very unusual uptake of cadmium compared to corn and soybeans and wheat. And I have never seen a report of high cadmium in tea or coffee. And it's just the nature of the transporters and the roots in the fruits.
Yeah. Okay.
okay, thank you.
Yeah, I'm happy about that myself.
That's why I was asking, because
I don't want to imply that chocolate is a risk. Okay, we talk with rice, 400 grams a day of dry rice.
Yeah.
How many grams of chocolate do you eat a day? Do you eat 100 grams once a week, once a month? Do you eat 5 grams a day? I mean, it's the amount of consumption and the bioavailability of the cadmium in the food that lead to absorption and risk.
Yes. Yeah.
So in my opinion, I mean, you should understand that because of the EU limit, industries in the US are forced to comply with that same kind of number. Food and Drug hasn't set an absolute limit, but California has, and that's where my consulting is right now with a court dealing with a case in California. But in Europe, it's absolutely based on cadmium alone. And every manufacturer does what they need to do. What they do is they mix poor flavor African cocoa beans with strong flavor South American, Central American cocoa beans to get the right taste and meet all the cadmium limits.
Yeah. Well, any other questions for Rufus?
Yes, I have a question.
Yes, please.
In some data, Rufus presented cadmium 109. This is a radioactive element. What equipment did you use?
I didn't hear the end of that. What?
Cadmium
Okay, I used,
109.
yeah, I did two kinds of experiments. One with 109, and that's gamma ray. And so I used a scintillation counter. And with the second one, we used, I think it's 112 or something like that, cadmium, which is you measure with a ICP MS. And we could, because we added enough of the stable isotope to shift the isotope ratio in the amended soil, we could readily measure the amount of cadmium from the biosolid versus background.
Okay.
There's a lot of new research using stable isotopes with very expensive mass spectrometers. You just do the search on stable isotope cadmium in wheat, and you'll find the introduction to that area of work.
Yeah.
MGA Mexico hosted in Geo 23 give us thank you for this presentation, everybody, and in special for Rufus. Thank you.
Thank you, Luis. And any additional questions for Rufus?
Bueno,
Well, I want to let everyone know that this presentation, like the previous ones, they have been recorded. Okay, so the presentation will be available on our consortium website, as well as it will be available also in YouTube and on our Facebook page as well. So if you want to go back and listen to this presentation one more time, or to the other ones that we have, the other speakers that we have invited into this speaker series, then you will be able to do so by just visiting our consortium website, or the Facebook page of the consortium, or the YouTube channel for the consortium. Also, materials that the speaker provide us, like Rufus has provided us with manuscripts that he has published. Those manuscripts also will be available for those of you that would like to get a copy of those articles.
Also, because I was government worker, most of my papers can be downloaded from ResearchGate. Just go to ResearchGate and look up Rufus Chaney, and you'll see a lot of papers that will give you information.
So Rufus, one more time, my dear friend, I would like to thank you for taking your time and sharing with us this fascinating research that you have done and your contributions to this field. This is a really very important area of work. And based on this research, we have had many improvements in soil amendments, in soil and the way that soils are being monitored, and also the quality of soils, and how we address the issue of toxic elements like cadmium and lead. So again, we thank you for giving us the honor and the privilege of having you here as one of our speakers for this seminar series.
Best wishes for your program. I enjoyed being able to help. I hope that, again, making it available on YouTube, etc., will let other people find this information.
Yes. Yes, it will be. We will send everyone. Jesus Ochoa is our consortium member that is in charge of facilitating this international speaker seminar series, and also facilitating all the different information that we have in our Facebook page and the YouTube channel. So I know that Jesus will send an email to everyone to let them know how to download or to look at the materials. So thank you everyone. Teresa, you have something to?
Yes, I wanted to make one last remark because as a chemist, and we do in our department a lot of analytical toxicology. I want to congratulate and to be thankful for all the huge analytical chemistry and work and the sampling that is behind all this wonderful research, because without proper sampling, storage, transport, and finally analysis, we wouldn't come to all these clues and results. And that is something we also highlight in our toxicology courses, because it's a faculty of chemistry. Therefore, thank you very much again, because this is very difficult to perform properly, we know.
I'll agree very strongly with this point. In the case of lead, laboratories had so much contamination from aerosol dusts in the cities, from gasoline and heating, that there was no correct lead analysis unless it was done in super clean rooms. Then by 1990, 1995, we didn't have lead in gasoline anymore, so it wasn't so bad. But it's only when, you're right, it's only when you use clean conditions to do your sample preparation and analysis, you can get honest values of lead.
Well, thank you very much. And thank you very much for Jose to announce and organize all this and introducing our speaker today.
Thank you. So on behalf, Rufus, one more time of the consortium and the International Medical Geology Association, we would like to express our appreciation for this wonderful presentation.
It was fun, and I hope it helps a lot of people.
Yeah, and I hope that you will be able to link to other presentations in the future as we bring other speakers as well. So you will be able to join us in the future as well. Okay.
Thank you.
So thank you, everyone, and
Thank you.
wishing everyone a wonderful weekend, relaxed weekend, and we see you next time during our next seminar, which should be in a month or so. Okay. So thank you, everyone. Bye-bye.
Bye. Thank you, Jesus, for your recording and all that. Bye.
Bye.
Bye-bye, Jesus.
Well.